following microbe is responsible for the production of blue cheese? Option A, Streptococcus thermophilus. Option B, Lactobacillus bulgaricus. Option C, Penicillium roqueforti. And option D, Rhizopus stolonifer. So the microorganism which is responsible for the production of blue cheese, it is Penicillium roqueforti. Even it provides a characteristic flavor and the texture to the blue cheese. Next question, number 2. Bacterial cell grown on hydrocarbon waste from the petroleum industry are a source of option A, carbohydrates, option B, proteins, option C, vitamins, and option D, fats. So the bacterial cells which are grown on the hydrocarbons, on industrial wastes or the byproducts, so these are the source of proteins. Option number B. Question number 3. Yeast cell crops harvested from the vats are used to produce which of the following compounds? So option number A, alcoholic beverages, option number B, enzymes, C, antibiotics, and D, organic acids. So the yeast cell crops which are harvested from the vats, they are used to produce the alcoholic beverages. That is option number A. Actually, yeast, it is a single cell microorganisms that can ferment the sugar to produce this alcohols, carbon dioxides. So next, what is the range of protein content in yeast cell? Option A, 69%, B, 12 to 15%, C, 20 to 40%, and D, 40 to 50%. So the range of protein in the yeast cell is around 40 to 50%. That is option number D. Number 5. For which of the following microorganisms have high vitamin content? Option A, bacteria, option B, yeast, option C, algae, option D, protozoa. So the following microorganisms which have a high vitamin content is yeast. That is option number B. Question number 6. The principal microorganisms for yogurt is option number A, Streptococcus thermophilus, option number B, Leuconostoc citrovorum, option number C, Lactobacillus acidophilus, option number D, Streptococcus lactis. So the principal microorganisms which is used in the production of yogurt is Streptococcus thermophilus. That is option number A. Even Streptococcus thermophilus, ये एक microorganisms, even two microorganisms are responsible. That the one more is Lactobacillus bulgaricus. यहाँ पे option नहीं दिया है, but for your information you can note it down. Even ये जो bacteria है, they are added to milk, and then the milk, it will ferment and it will produce a lactic acid, and which will cause the milk to coagulate and form a thick tangy yogurt. So here the correct option is option number A. Question number 7. Which of the following products have higher acidity and lacks aroma? Option A, cultured buttermilk, option B, cultured sour cream, option C, Bulgarian milk, and option D, acidophilus milk. So the product which have higher acidity and lacks aroma is option number B, that is cultured milk. So cultured milk, actually it is made by fermenting the cream with lactic acid bacteria. So the product it produces is lactic acid, and even it lowers down the pH of the milk. Whereas this cultured buttermilk, it is a fermented dairy product which have a tangy flavor or aroma. And number C, that is Bulgarian milk. Bulgarian milk, it is a type of yogurt which is known for its creamy texture, and even it also has a mild flavor. And option number D, that is acidophilus milk, it actually it is a probiotic, and it is known for its health benefits. Even it also has a mild flavor, aroma. Question number 8, that is shredded cabbage is the starting product for which of the following fermented food? Option A, sauerkraut, option B, pickles, option C, green olives, and option D, sausages. So the correct option is option number A, that is sauerkraut. Actually, shredded cabbages, they are fermented, and this fermented product, it is known as sauerkraut. Question number 9, most spoilage bacteria grow at option A, acidic pH, option B, alkaline pH, option C, neutral pH, and option D, any of the pH. So most spoilage bacteria, they grow at option number C, that is neutral pH. So it ranges from actually pH 5 to pH 8. Question number 10. Suspected colonies of Staphylococcus aureus when grown on bed Parker medium shall show option A, coagulase activity, option B, protease activity, option C, catalase activity, and option D, none of these. So the suspected colonies of Staphylococcus aureus, when it is grown on the Baird-Parker medium, it actually shows coagulase activity. That is option number A. So this Baird-Parker medium, agar, it is actually a selective as well as a differential medium which is used for the isolation as well as the enumeration of coagulase positive Staphylococcus in the food. So my correct option is option number A. Question number 11. Which of the following acid will have a higher bacteriostatic effect at a given pH? Option A, acetic acid. Option B, tartaric acid. Option C, citric acid. And option D, malic acid. So the bacteriostatic effect, it actually depends of an acid actually depends on the two factors. One on the concentration and another on the pH of the environment. So however, the acetic acid, it has a higher bacteriostatic effect than other of the following acids which are given. So so my correct answer is option number A, that is acetic acid. So this acetic acid actually, it is a weak organic acid that is commonly used as a food preservative due to its antimicrobial properties. Even it is effective against a wide range of gram positive as well as gram negative bacteria. And this tartaric acid, citric acid, malic acid, these are also used in food industries, but they are not as effective as the acetic acid. So here the correct option will be option number A, that is acetic acid. So question number 12. Which of the following is not true for the thermal resistance of the bacterial cell? Option A. Cocci are usually more resistant than rods. Option B. Higher the optimal and maximal temperatures for growth, higher the resistance. Option C. Bacteria that clump considerably to form capsules are difficult to kill. And number D. Cells low in lipid content are harder to kill than other cells. So the correct option is option number D, that is cells low in lipid contents are harder to kill than other cells. In fact, in this, the cells which are high in lipid content such as these spores, so they are more resistant to heat than the cells which are low in lipid content. So here the statement which is not true is number D, that is cells low in lipid content are harder to kill than others. Question number 13. Water activity can act as option number A, an intrinsic factor determining the likelihood of microbial proliferation. Option number B, a processing factor. Option C, an extrinsic factor. And option number D, all of the options. So the correct answer is option number D, all of the above. So what does actually water activity mean? Water activity, it is a measure of amount of free water available in the food or other substances. It is actually a ratio of water vapor present in the food substance to that of the pure water at a given temperature, means same temperature and pressure. So this term is important, at same temperature and pressure. So the correct option is option number D, all of the above. So question number 14. Enumeration of microorganisms refers to option A, either spiral plating, pore plating or spread plate of a food suspension onto a suitable selective agar, or non-selective plate, depends on the test. Option B, either spiral plating, pore plate or spread plate of a food suspension onto a suitable selective agar. Option C is non-selective plating, depending on the test. Option D, none of the options. So actually enumeration of microorganism, it refers to either spiral plating, pore plate or spread plate of a food suspensions onto a suitable selective agar or non-selective agar, depending on the tests. So the correct option is option number A. Question number 15. The different aerobic colony counts between food categories reflect the option A, expected level of contamination of the raw material. Option B, potential for microbial growth during storage. Option C, potential shelf life. And D, all of the above. So the correct answer is D, all of the above. So this different aerobic colony count, it depends on various conditions. Even other conditions can be the type of food, processing of the food, its storage conditions. So all of the above is the correct answer. Next. Question number 16. Examination of the presence or absence of the microorganisms refers to option number A. Incubation of food suspension in an enrichment medium followed by incubation onto a suitable selective medium. B. Incubation of food suspension in an enrichment medium followed by incubation onto a non-selective medium. Option C. Enrichment of food suspension in an enrichment medium, incubation of food suspension in an enrichment medium, and option number D, none of the options. So the correct answer is option number A, that it is incubated the food suspension in an enrichment medium and then it is followed by incubation onto a suitable selective medium. Question number 17. Question number 17. Plate count of bacteria in foods generally use the plating medium consisting of option A. Peptone, yeast extract, glucose, sodium chloride, agar, and distilled water. Number B. Yeast extract, glucose, sodium chloride, agar, and distilled water. Peptone, glucose, sodium chloride, agar, and distilled water. D. Peptone, yeast extract, glucose, sodium chloride, agar, and distilled water. So the correct option is option number A, that it will contain of peptone, yeast extract, glucose, sodium chloride, agar, and distilled water. So this peptone and yeast extract, it is responsible for providing the nitrogen and other nutrients which will support the growth of the bacteria, whereas glucose, it will provide the source of energy to the bacteria, while sodium chloride, it will help to maintain the osmotic balance of the medium, and agar, it will act as a solidifying agent, as well as it will provide a surface for the bacteria to grow, and distilled water, it is used for the adjustment of the medium and pH, to adjust the pH. So option number A. Next, question number 18. What are the intrinsic factors for the microbial growth? Option A, pH, option B, moisture, option C, oxidation reduction potential, and option D, all of these. So the correct answer is option number D, all of these. So the pH of a food product, it can affect the growth of the microorganisms by altering the acidity of the environment, whereas moisture content can also affect microbial growth by providing suitable environmental conditions for the microorganisms to grow or for its reproduction. And oxidation and reduction potential, so actually oxidation and reduction potential, it is a measure or the tendency of the substance to gain or lose the electrons that can affect the microbial growth by altering the availability of the oxygen or the environment. So here the correct answer is option number D, all of these. Next, number 19. Yeast and mold count determination requires option number A, agar, nutrient agar, option number B, acidifying potato glucose agar, option C, McConkey agar, option D, violet red bile agar. So the correct option is option number B, that is acidified potato glucose agar. So the plating media which is used for the yeast or mold count, it is used for the determination of acidified potato glucose agar. So this medium is actually a selective for fungi, and it is used for the determination of the yeast and mold colonies which are present in the food sample. So here the correct answer is option number. Question number 20. The time temperature combination for HTST pasteurization. So what does HTST stands for? HTST stands for high temperature short time pasteurization of 71.1 degrees Celsius for 15 seconds is selected on the basis of option number A, Coxella burnetii, B, E. coli, C, Bacillus subtilis, and C, D, Clostridium botulinum. So here the correct option will be option number A, that it is Coxella burnetii. Question number 21. Aerobic colony count is also known as option A, total viable count, TVC, option number B, aerobic plate count, APC, option C, standard plate count, SPSC, and D, all of these. So aerobic colony count, it is also known as TVC, APC, and SPC. All of these is the correct answer. Question number 22. Which of the following is true about ISO 2002 method for salmonella detection? Option A, selenite cysteine SC broth is replaced by Muller-Kaufmann tetrathionate novobiosine broth, that is KMTTN. B, Rapaport Vaciliades RV broth has been replaced by Rapaport Vaciliades soya RVS broth. Option C, XLD is the first isolation medium rather than BGA. Option D, all of these. So the correct option for this will be option number D, all of this. Question number 23. A sacrophilic halophile would be a microbe that prefers option A, cold temperature and increased amount of salt, B, warm temperature and increased amount of pressure, C, cold temperature and the absence of oxygen, warm temperature and increased amount of acids. So actually sacrophile. Sacro means what? Cold, and philic means loving, that is cold loving. And halophile, halo means salt concentration and phile means loving. So the microorganisms which loves or prefers to be in a cold temperature as well as in a higher salt concentration environment. So here the correct option will be option number A, that is cold temperature and increased amount of salt. Question number 24. NaCl can act as option A, antagonist at optimum concentration, option B, synergistically if added in excess of optimum level, C, both A and B, and D, none of the above. So here the correct option will be option number C, that is both A and B. So the effect of NaCl on the microbial growth, it will depend on the concentration and the type of microorganisms. Say at low concentration NaCl, it can act as a growth stimulant for microorganisms, while at higher concentration it can act as a growth inhibitor. So the optimum concentration of NaCl for microbial growth, it varies depending on the type of microorganisms and the food products. So in some cases antagonist, the NaCl can act as an antagonist by inhibiting the growth of certain microbial or the microbes at optimum concentration, or in other cases it can act, that NaCl can act as synergistically while other factors to promote the growth of the microorganisms, such in case of the halophilic bacteria that requires a high salt concentration for its better growth. So here option number C is correct. Question number 25. The microbial examination of coliform bacteria in food preferably use option A, McConkey broth, option B, violet red bile agar, option C, eosine methylene blue agar, and option number D, all of these. So do comment down this correct option for this, and also comment down your score out of 25 which you have secured. So thank you everyone. You can subscribe our YouTube channel and other social media links. It is given in the description box. So till then, enjoy learning. Thank you.